Hello, welcome to Molbiolect. My name is Ankit Kumar Nath. Today's video will be based on viruses. What are viruses? And the classification of virus. The classical Baltimore classification I will be talking about. How you can remember them very easily. What is the trick? So first, let's get into the video. But before going into the video, I would just like you to know that you should go and subscribe to my channel. Press that red subscribe button and also the bell beside it, so that whenever I upload a new video, you will get the notification and you can learn fast. So let's start with the first portion of our video. So the first portion of our video I'll be dealing with is water viruses. So it's the first question over here. This is what is water viruses. Viruses are basically submicroscopic particles. So submicroscopic meaning they are not visible in a light microscope. They need electron microscope in order to identify them and in order to look at them. So these viruses needs a host, a live living host. So this living hosts provide them with everything that they need to multiply them, like we multiply ourselves. So viruses are basically dead when they're outside a host, but whenever they're inside a host, they are alive. Starting with, what do you mean by classification of viruses? So this is our second question over here. What do you mean by classification of virus? Classification of virus simply means that how you can divide the viruses according to different kind of aspects. So what are those aspects? So, and why do you need to classify? Okay, my third question over here, that why do you need to classify the viruses? The need for classifying is that there are many different kind of viruses, starting from one type to the another. So you need to group them into their respective portions and sub-portions. Okay, so this classification is done by a very common procedure, very known procedure to all of us, that is the Baltimore classification of viruses. Now this Baltimore classification of viruses deals with 7 groups. Okay, these 7 groups are 7 groups that are group 1 to group 7, and these are classified by categorizing their nucleic acid, their strandedness as positive or double stranded DNA, the sense of the DNA or the RNA, and the method of replication. These 4 classification aspects are taken and they are classified into 7 groups. So let's go into the Baltimore classification now. So we have here the Baltimore classification of virus, and this Baltimore classification of virus is in accordance with the 1971 model by David Baltimore. So we have the group 1 viruses. Now these group 1 viruses, let me get my camera a bit closer. Okay, I hope you can see it. So the group 1 virus are basically double-stranded DNA viruses, like the Adenovirus, the herpes virus, the pox virus. So we have these viruses over here which are under group 1. So they are having double-stranded DNA. The next I'll be talking about is the group 2 single-stranded DNA, which can be a positive strand or positive sense. Is like parvovirus, which can infect humans causing immunosuppression, very much the other opposite to the SARS-CoV virus. We also have the group 3 dsRNA viruses. So I'm talking about the group 3 dsRNA viruses. These viruses are basically the reovirus. Okay, we have the reovirus which causes subclinical manifestations, and these manifestations are basically in human, so we don't get so much sick because of this, and it is basically treatable, so we don't get this virus into so much consideration nowadays. We also have group 4 viruses, single stranded RNA, which are positive sense, obviously our very famous coronavirus right now, along with picornavirus and toga viruses. These are all basically on the same group, that is group 4. These are RNA viruses. Remember one thing, group 3, group 4, group 5, group 6 and group 7, that is group 3 onward, everything is RNA virus. So group 5 we have the single stranded RNA, which is of negative sense. Example is orthomyxovirus and rhabdovirus. So in generally in your course you will need to remember actually group and one example at least, and the type of the genome, that is positive stranded, single stranded DNA, double stranded DNA. This you have to remember. Also we have got the group 6, okay, group 5 ssRNA minus sense, orthomyxo and rhabdo. Group 6 we have single strand RNA, reverse transcriptase viruses. Okay, positive strand or sense, anything you can say, RNA with DNA intermediate in life cycle. So basically these viruses contain RNA as their genome, but once they enter the cell, they convert their RNA into DNA, and this is helping in their further life cycle. The next we have here is group 7, that is the dsRNA-RT reverse transcriptase viruses. So it also contains the enzyme reverse transcriptase over here. So the example is, so basically it also contains DNA with an RNA intermediate. So the virus has a DNA, but it is present in an RNA intermediate form inside the host, and this helps in its life cycle. Example, the HEPA-DNA viruses. So this is all group 1 to group 7. Now my question is, how can you remember this? Okay, how can you remember this very easily? Because group 1 to group 7, so many examples, so many types, single strand, double strand, positive sense, and all. You have to remember this. So how can you remember? Just note a simple thing. You all know about music, classical Indian music. Okay, so there is something like rag or something, I don't know about that, but this positive strand, this dsDNA, ssDNA can be very easily remembered by using a musical note like dhin ta 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 dhin. Okay, like a tabla tuning, dhin ta 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 dhin. So here dhin means the dsDNA and ta means the ssDNA. So dhin ta 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 dhin is basically how you are converting you are actually representing the double stranded and single stranded DNA very easily. Now a question may come that how can we actually understand that what is the sense of the strand, the sense of the positive strand or negative strand? How can we identify that with the help of this this thing I have just shown here, dhinta and all? Very simple. You have to remember that, or you can notice it over here, that all the even numbers 2, 4, 6, these all contain positive sense genomes. Okay, RNA or DNA you are getting from this thing I have shown over here. So you're getting from this thing that is RNA or DNA. Okay, but the positive or negative strand, very simple. All 2, 4 and 6 contains positive strand. Even one is positive strand. Negative strand only group 5. So we have only group 5 over here, which is having the negative strand. My highlighter is not working properly. Okay, it's working now. Now, apart from that, how can we remember the examples like at least one examples like adenovirus, parvovirus, reovirus? So I have got a synonym. Okay, synonym or acronym, I don't know what you call it. Simile, I guess, I don't know. Okay, simple sentence which can help you remember every example. So the synonym is that, and you have to notice it. So it is Adam was pervert, but Rio was together with Corona beard, learning orthomixing retro music at Hepadna Cafe. So Adam was pervert, but Rio was together with Corona beard, learning orthomixing retro music. So I don't know what is orthomixing, I don't know. Retro music I know. At Hepadna Cafe. So cafe's name is Hepadna. Adam and Rio were together over there, and they were having Corona beer, but Adam was pervert and also Corona beer, but Rio was learning how to orthomix, or basically mix some kind of retro music into modern music form, maybe at Hepadna Cafe or Hepadna Cafe. Okay, so this is how you can remember the whole of the Baltimore classification in just 1 minute. Okay, first is the musical note dhinta tata dhin, followed by the positive negative using 246, sorry, not 8, and the whole example can be remembered by using this simple sentence. Now let's talk about the RNA viruses itself. Okay, so what are these RNA viruses? What good does they do? Are they helpful? Are they harmful? So let's learn about them. Now basically RNA viruses are group 3, group 4, group 5, group 6 and group 7. So group 3 viruses have a positive sense single stranded genome. Obviously RNA, because I'm talking about the RNA viruses. Example is rotavirus. Group 4 poses a positive sense single stranded RNA genome, for example picornavirus, hepatitis A virus, enteroviruses. And all these viruses like the rotavirus and everything, they need specific host tissues which are actively dividing. Also we have group 5 negative sense ssRNA that are deadlier than SARS. okay, Ebola virus, Marburg virus. These are deadlier than SARS, and also some less deadlier like influenza virus. Group 6, we have got single-stranded RNA that replicate through DNA intermediate. Example, the HIV, the thing you do not get by sitting on toilet seat. Okay, just let's get this thing clear. Also, we have got group 7 has DNA genomes, double-stranded DNA genomes, but they replicate using reverse transcriptase. Example, the hepatitis B virus. So this is all the RNA viruses that we know. Okay, and out of this, the group 4 contains our very own SARS-CoV2 virus, which is mutating at a very fast rate. Discovering vaccines is getting more and more painful right now. Also, just I would like to say that if I get a good response in this video, okay, if you guys subscribe more, if you guys share this more, also if you guys like my video and comment down below, then I will make another video which deals with LHT systems of viral classification. It's a much newer system, it's much defined system than Baltimore system. But Baltimore is very commonly used everywhere. Now, this ends our second part of the video. Now we come to third part of the video. So, we have got this SARS-CoV2 outbreak and we are searching for drugs. But do we have any other alternatives other than drugs? Not vaccines I am talking about. Are there anything that are made out of the human cells only and they are very much potent in curbing down the symptoms of this SARS-CoV2? I think we have, and this is the PLX cells. The PLX stands for the pluristem cells, and these pluristem cells are basically allogenic. So we have this term over here, allogenic. So what is allogenic? Allogenic means they are from the same species. For example, if I graft your skin, so suppose I am taking my skin and grafting it in yours. So the species is same, but your genetic nature and my genetic makeup will be entirely different. But the fact is that my skin grafting, my skin portions will be non-invasive to your body, and your body would not actually identify it as a threat. Basically, what we try to do in any kind of transplants, okay, grafting transplants and everything. So they are these PLX cells are basically allogenic mesenchymal cells that have immunomodulatory properties. Now, what is this term immunomodulatory means? Immunomodulatory means that whenever you are trying to suppress the immune system, or rather you are trying to say your immune system that it is not the actual threat. So basically it's modulating the immunity, it's controlling your immunity, it's regulating your immunity. So these are immunomodulatory properties. They induce the T regulatory cells. So these T regulatory cells, what they do is that I have already written, they suppress the immune cells. They suppress the immune activation. And also M2 macrophage. It secretes arginase 1, IL-10, TGF-beta and anti-inflammatory cytokines. So I was talking about viruses. So why am I coming to this kind of immunosuppressive things, therapies that I am trying to provide? That why I'm talking about this? Basically, you have to understand, SARS-CoV2 is itself not that much dangerous. What makes it dangerous is because it is overactivating your immune system. Your immune system identifies it as a threat. It's very good, but it is overactivating itself. The SARS-CoV2 helps you to overactivate your immune system, and also it tries to block many kind of interferons and everything. So we need to understand this. Your immunity is against yourself right now, like an autoimmune disease. Okay, so what happens that's the sole purpose that many autoimmune drugs are also included in the trial. But why am I talking about this? So if I induce more and more T regulatory cells and M2 macrophages, what it will cause is that it will suppress your immune cells, and it will help you prevent an essential symptom of this kind of virus attack, and that is pneumonitis, where your lung cells are so much inflamed that it turns pneumonic. Okay, there is overactivation and there are highly inflamed lung cells present, and these lung cells are structurally so much damaged that whenever you are passing an X-ray beam to get a lung X-ray, the X-ray thing is completely penetrating and gives you white spots in the X-ray film. I will add a photo at the last of this video, at the end of this video. So this is how using pluristem PLX cells, that are allogenic mesenchymal cells, we can actually induce the suppression of your immune system. So not induce, actually suppression of your immune system, and this can be done by using the process of lung grafting. Okay, so we are going to graft a tissue of this kind of cells in your lung, or we are going to artificially inject it into your lungs, and these live tissues will actually make you less overactive in your immune sense, such that you do not kill yourself, overactivating your immune self or immunologic properties. So this covers my entire video. So this was the focus of my video today, that what is Baltimore classification, what are viruses in details, and also the 7 groups, the RNA viruses, and do we have anything other than drugs that can be useful in treating this kind of SARS-CoV-2? So the answer is yes. But the question comes, where is this process invented? Basically invented in the Jerusalem. Okay, so very much interesting paper. I have gone through the whole paper. There are many things. Clinical trial is going on with this. They have found very good results, and also this is a bit costly process, but if it is developed in a proper scale, I think we should also, the government will try to reduce its costing by regulating some kind of other processes, and also by making some kind of grants and all everything. I don't know about that, but this thing actually works very good according to their paper. Okay, so by saying this, I would just like to say that you should go down in my channel, subscribe, press that red subscribe button, it will turn gray, it's a magic, and also please press that bell icon. Whenever you are pressing that bell icon, it actually helps you to get the most accurate information fastest possible from me. And also you should go through all these papers and all these things. You should Google this, the PLX cells, everything is in Google. If you Google it, you will get everything. And regarding Baltimore classification, this whole thing that the dhinta thing over here, you will not going to get it anywhere. It's only in my channel, and I will also provide you many different kind of informations and this kind of things which will make you easy to learn every tough and difficult topic. So please subscribe to my channel. Okay, that's very much essential. For now, Molbiolect out. So I'm concluding this video by saying this. Bye, and we will later meet at my next video, where I will be talking about another issue which is now suppressed, kind of suppressed, but it's also a very challenging issue we are facing, that is antibiotic resistance of the bacterial cells, the multi-drug resistant cells. Okay, so I've already uploaded a video related to conjugation, but I will also cover other aspects in more details. Okay, so please like, share, comment, and subscribe to my channel, and be notified by pressing that bell icon. Bye.